Ready to go. Today we will be discussing the third chapter in the book of cellular immunology, and we will be discussing this in detail for the coming time that I cannot really approximate, but let us go together. So, chapter 3, leukocyte circulation and migration into tissues. So in this chapter, we'll look into overview of leukocyte migration, adhesion molecule on leukocyte and endothelial cell involved in leukocyte recruitment, selectins and selectin ligands, integrins and integrins ligands, chemokine and chemokines receptors, chemokine structure, production and receptors, function of the chemokines, and other chemoattractant and receptors. Because leukocyte endothelial interaction and leukocyte recruitment into tissue will be looked, migration of neutrophil and monocyte to sites of infection or tissue injury. We'll look on migration and the recirculation of T lymphocyte, recirculation of naive T lymphocyte between the blood and secondary lymphoid organs, recirculation of T lymphocyte through the other lymphoid tissue, migration of effector T lymphocyte to tissue of infection, memory T cell migration, migration of B lymphocyte, whereby look to recirculation of naive follicular B cell, migration of antibody secreting plasma blast and memory B cell. Then we look on to summary. So let us start. A unique property of immune system that distinguishes it from all other physiological systems in the body is the constant and highly regulated movement of its major cellular component through the blood into tissues and often back into the blood again. This movement accomplishes three main functions. Number one, delivery of leukocyte of myeloid lineage, mainly neutrophil and monocyte, from the circulation into the tissue site of infection or injury, where the cells perform their protective function of eliminating infectious pathogen, clearing dead tissues, and repairing the damage. Secondly, delivery of lymphocyte from their site of maturation, this is bone marrow or thymus, to secondary lymphoid organ, where the cell recognize antigen, proliferate, differentiate into effector and memory lymphocytes. Third, delivery of effector lymphocyte from the secondary lymphoid organs in which they were produced to site of infection in any tissue where they perform their protective function. Because of the ability of immune cells to disseminate throughout the body, an immune response may be initiated at one site but may be active at distant locations. In other words, immunity is both local and systemic. The migration of a leukocyte out of the blood and into a particular tissue or to a site of infection or injury is often called leukocyte homing, and the general process of leukocyte movement from blood into tissue is called migration or recruitment. The ability of lymphocyte to repeatedly home to a secondary lymphoid organ, reside there transiently, and return to the blood is called recirculation. The recruitment of leukocyte and plasma protein from the blood to the site of infection and tissue injury is the major part of the process of inflammations. Inflammation is triggered by recognition of microbes or injured or dead cell in innate immune response, and it is refined and prolonged during adaptive immune response. The inflammatory response delivers the cell and molecule of host defense to the site where the offending agent need to be combated. The same process is responsible for causing tissue damage and underlying many important diseases. We will return to the inflammation in the context of innate immunity in chapter 4 and in the discussion of inflammatory disease in chapter 19. Overview of leukocyte migration. Leukocyte homing and recruitment to different tissue are governed by some general principles. First, naive lymphocytes continuously migrate mainly into secondary lymphoid organ, where the lymphocytes that have been previously activated by antigen, e.g. effector lymphocytes, as well as myeloid leukocytes, preferentially home into tissue where there is an infection or tissue injury. Memory lymphocytes migrate into lymphoid organ, mucosa tissue, skin, and other tissue. You can see the diagram which is showing the lymphoid and other organ in this part. So in the diagram, as you'll see in the picture, it explains more detail on the function served by leukocyte migration from the blood tissues. So other function is leukocyte homing and recruitment require adhesion of leukocyte to the endothelial lining of postcapillary venules, a process that involves molecules on the surface of both leukocyte, this adhesion molecules, and chemokines receptor, and endothelial cells, adhesion molecule, and chemokines. Endothelial cells at the site of infection and tissue injury are activated by cytokines secreted by sentinel cells in the tissue, including dendritic cells, macrophage, and mast cell, which induce increased expression of adhesions molecule and chemokines. The consequences is increased adhesiveness of the endothelial cells by recirculating lymphoid leukocyte and previously activated lymphocyte. Because microbes and necrotic tissues stimulate the expression of molecules that mediate leukocyte endothelial adhesions at the site of infection or injury, effector leukocyte migrate through endothelium mainly when and where they are needed. The basic process of leukocyte migration into tissue is the same for the homing of different types of leukocyte. This is neutrophil, monocyte, and naive and effector lymphocytes. To differentiate types of tissue, this is secondary lymphoid organ infected tissue. Although these specific chemokines and adhesion molecules vary in the way that result in distinct pattern of migration of different cell types, before describing the process, we'll discuss the properties and function of adhesion molecule and chemokine that are involved in leukocyte recruitment. Adhesion molecules and leukocyte endothelial cells involved in leukocyte recruitment. Adhesion of circulating leukocyte to vesicular endothelial cell is mediated by two classes of molecule called the selectin and integrin, and they are ligands. The expression of these molecules varies among different types of leukocyte and in blood vessel at different locations. Selectin and selectin ligands. Selectin are plasma membrane carbohydrate binding adhesion molecule that mediate an initial step of low affinity adhesion of circulating leukocyte to endothelial cell lining postcapillary venules. The extracellular domain of selectin are similar to C-type lectins, so called because they bind carbohydrate structure. Lectin are carbohydrate binding proteins in a calcium-dependent manner. Selectin and their ligands are expressed on leukocytes and endothelial cell. Importantly, the expression of selectin and their ligand is increased by exposure to cytokine produced at site of tissue injury and infection, and this is one of the mechanisms by which leukocytes are recruited to the site of inflammation. Endothelial cells expresses two types of selectins called P-selectin, CD62P, and E-selectin, CD62E. P-selectin, so named because of the first found in platelet, is stored in cytoplasmic granules of endothelial cells and is rapidly redistributed to the luminal surface in response to histamine from the mast cell and thrombin generated during blood coagulations. E-selectin is synthesized and expressed on endothelial cell surface within 1 to 2 hours in response to cytokine interleukin-1 and tumor necrosis factor TNF, which are produced by tissue sentinel cells, the dendritic cell, and macrophage in response to infection. Microbial products such as lipopolysaccharide (LPS) are also stimulates E-selectin expression on endothelial cells. We will describe the interleukin-1, tumor necrosis factors, and LPS in our discussion of inflammation in Chapter 4. The ligand on leukocytes that bind to E-selectin and P-selectin on endothelial cells are complex ciliated carbohydrates related to the Lewis X and Lewis A family of blood group molecules. These chemical structures are present on various surface glycoprotein of granulocyte, monocyte, and some previously activated effector and memory T cells. The best defined of these is the tetrasaccharide salcyl Lewis X. The structure was originally identified as an antigen on the red blood cell in studies of genetically related people with the family names Lewis. A leukocyte membrane glycoprotein called P-selectin glycoprotein ligand 1, PSGL1, is post-translationally modified to display Cl Lewis X, which serves as the major carbohydrate ligand for P-selectin. Several different molecules may display the carbohydrate ligand for E-selectin, including the glycoprotein PSGL1 and E-selectin ligand 1 and some glycoprotein. A third selectin called L-selectin, CD62L, is expressed on leukocytes and not on endothelial cells. The ligand for L-selectins are sialomucins on endothelial cell, whose expression may be increased by cytokine activation on cells. A major recognition determinant that L-selectin bind to on these sialomucin is Sil-6-sulfur Lewis X. The expression of the ligand on endothelial cell is increased by interleukin-1, tumor necrosis factor, and other inflammatory cytokine. So L-lectin or neutrophil promote the adhesion of these cells to the endothelial cell and site of inflammation. In adaptive immunity, L-selectin is required for naive T and B lymphocyte homing into lymph node through a specialized blood vessel called high endothelial venules, HEVS. The ciliated blood vessel called the high endothelial venule, the HIVS. The sialomucin ligand on high endothelial venules that bind to L-selectin on naive lymphocyte are collectively called peripheral node addressing, the PNAD. Leukocyte express L-selectin and carbohydrate ligand for P-selectin and E-selectin at tip of their microville, facilitating interaction with molecule on endothelial cell surface. Integrins and integrin ligands. Integrin are cell surface protein that mediate adhesion of the cell to other cells or extracellular matrix through a specific binding interaction with various ligands. There are more than 30 different integrins, all of which are heterodimer, containing one of more than 15 types of chain and one of the 7 types of B chains. The extracellular globular head of both chain contribute to ligand binding. The cytoplasmic domain of integrin interact with the cytoskeletal component, including vinculin, taurine, actin, alpha-actinin, and trypomycin. The name integrin for this family of the protein derived from the idea that this protein integrate signal triggered by extracellular ligand with cytoskeletal dependent motility, shape change, and phagocytic response. Although we will focus on leukocyte adhesion function of integrin, these molecules have many other biological functions, including adhesion, proliferation, and differentiation of epithelial cell, and platelet adhesion and aggregation during blood coagulation. Inflammatory cytokines and other signal increase endothelial expression of integrin, ligand and binding affinity of leukocyte integrin, thus promoting leukocyte adhesion to endothelial at site of inflammation. In the immune system, two important integrins that are expressed on myeloid cell and lymphocyte are leukocyte function-associated antigen, LFA1, more preciously named as alpha L beta 2 or CD11A CD18, and very late antigen 4, VLA4, also named as alpha 4 beta 1 or CD49D CD29. See more table on table 3.1. One important ligand for function-associated antigen, LFA1, is intercellular adhesion molecule 1, ICAM1, CD54, a membrane glycoprotein expressed on cytokine-activated endothelial cells and on variety of other cell types, including lymphocyte, dendritic cell, macrophage, fibroblast, and most epithelial cells. The extracellular portion of ICAM1 is composed of globular domain called the immunoglobulin IG domains, which share sequence, homology, and structural features with the domain found in Ig molecule. Many proteins in the immune system contain Ig domain and belong to Ig superfamily. LFA1 binding to ICAM1 is important for leukocyte endothelial interaction, discussed later, and T cell interaction with antigen presenting cell, APC, seen chapter 9. Two other Ig superfamily ligands, the LFA1 and ICAM2, which is expressed on endothelial cell, and ICAM3, which is expressed on the lymphocyte, VLA4 bind to vesicular cells adhesion molecule I1, this is VCAM1, CD160, and Ig superfamily protein expressed on cytokine activated endothelial cell in some tissues. Other integrins also play roles in innate and adaptive immune response. For example, MAC1, alpha M beta 2, CD11, CD18, CD11B, CD18, or complement receptor 3, CR3, on circulating monocyte bind to ICAM1 and mediate adhesion to endothelium. MAC1 also functions as complement receptor, as does another integrin of beta 2 family known as CD11C CD18, the alpha beta complement receptor 4, CR4. Both bind particle opsonize with the product of complementary activation called the inactivated C3b IC3b fragment, discussed in chapter 4 and 13, and thereby enhance phagocytosis of microbes. CD11C CD18 is expressed mainly on dendritic cell, but also in other myeloid cells and activated B cell. It helps dendritic cell ingest the apoptotic cell. The integrin alpha 4 beta 7 is expressed on lymphocytes that home in intestinal mucosa and bind to an endothelial protein called the mucosa addressing cell adhesion molecule, the MADSCAM1, alpha E beta 7 CD103, is an integrin that bind to an epithelial adhesion molecule called E-cadherin. Alpha EB7 is expressed on subset of T cell and D cells that are found within epithelial layer of mucosa. Integrin rapidly increase affinity for their ligand in response to intracellular signal, which are induced in all leukocytes by chemokine binding to chemokine receptor and in T cell by antigen binding to antigen receptor. Chemokine receptor and antigen receptor engagement trigger signaling pathway described in more detail chapter 7 that lead to the association of RAP family and small GTPase protein and cytoskeleton interacting protein with the cytoplasmic tail of integrin protein. This results in conformational changes in the extracellular domain of integrin that lead to the increased affinity. In the low affinity state, the stack of extracellular domain of each integrin subunit are bent over and the ligand binding a globular head are close to the plasma membrane. In response to alteration in the cytoplasmic tail, the stack extend, bringing the globular head away from the membrane to a position where they are more effectively interacting with their ligand. The process by which intracellular ligand generated in response to chemokines or antigen alter the binding function of extracellular domain of integrin is called inside-out signaling. Chemokine also induce clustering of integrin or leukocyte surface. This results in higher local concentration of integrin at site of interaction with endothelial cells, where the chemokines are displayed, leading to increased overall strength ability of integrin-mediated leukocyte binding to the endothelium. Chemokine receptor. Chemokine are a large family of structural homologous cytokines that stimulate leukocyte movement and regulate the migration of leukocytes from the blood to the tissue. Chemokines stimulate the movement of leukocyte along the concentration gradient of the secreted protein toward their source, a process called chemotaxis or chemoattraction. The name chemokines is a contraction of chemotactic cytokines. We referred to the role of chemokines in the organization of lymphoid tissue in chapter 2, and now we'll describe the general properties of this family of cytokines and their properties, their multiple function in innate and adaptive immunity. Summary is found in table 3.2, where they summarize the major features of selecting chemokines and their process. Chemokine structure, production, and receptor. Most chemokines are 8 to 10 kilodalton polypeptides that contain 2 internal disulfide loops and 4 cysteine residues that are responsible for tertiary structure. There are about 47 human chemokines, which are classified into 4 families on the basis of the number and location of 2 of the conceptive systems. The 2 major families are CC, called beta chemokines, in which 2 defined cysteine residues are adjacent, and the CXC or alpha chemokine, in which these residues are separated by 1 amino acid. In both mice and humans, the gene encoding most of the CC or CXC chemokines are found in distinct clusters on different chromosomes. A few additional chemokines have either a single cysteine, C family, or 2 cysteines separated by 3 amino acids, CX3C. There are 2 structural variants of CXC chemokines. Some have the amino acid sequence glutamic acid leucine adenine, called ELR motif, just before the first cysteine of CXC motif, and other without ELR motif. CXC chemokines with ELR motif support neutrophil migration. The other CXC chemokines and CC chemokines act on monocyte, lymphocyte, and other leukocyte. Chemokines were originally named on the basis of how they were identified and what response they triggered, but a standard nomenclature has been adopted and coordinated with names of receptors and chemokine bind to. The CC chemokines named as CCL1 through CCL28 and CXC chemokines are named CXL1 through CXL17. Chemokine bind tightly to glycoaminoglycan with sulfide N-acetyl-stannamines, disaccharide repeat unit, and there is specificity for which chemokine bind depending on the length of their chains of repeat and pattern of sulfation. These glycoaminoglycan on the surface of endothelial cells or in extracellular matrix display the bound chemokines to chemokines receptor expressed on migrating cells. Chemokine of CC and CXC subfamily are produced by leukocyte and by several types of tissue cells such as endothelial cells, epithelial cell, resident macrophage, fibroblast, and other stroma cells. In many of these cells, secretion of chemokines is induced by recognition of microbes through various cellular receptors of innate immune system discussed in chapter 4. In addition, inflammatory cytokines including tumor necrosis factor, interleukin-1 and interleukin-17 induces chemokines production also. Several CC chemokines are also produced by activated T cells, providing a link between adaptive immunity and recruitment of inflammatory leukocytes. So there is a table selected chemokine and chemokine receptor, which you can have a look at it. But in summary, we have CC chemokines by start. So like CCL2, original name is MCP1. Chemokine receptor is CCR2. They are found in mixed leukocyte recruitment, and their function is main in mixed leukocyte recruitment. CCL3, CCR1, CCR5, mixed leukocyte recruitment also, the main function. CCL4, the receptor is CCR5. They are mostly found in T cells, dendritic cells, monocyte, and natural killer cell recruitment, HIV co-receptors, the CCR5. CCL5, the chemokine receptor is CCR1, CCR3, and CCR5. They are found and the main function is mixed leukocyte recruitment. CCR11, the chemokine receptor is CCR5, R3. Their main function is eosinophil, basophil, and Th2 recruitment. CCL17, the chemokine receptor is CCR4. They are found in, the main function is T cell recruitment. CCL19, their chemokine receptor is CCR7. These the main function in T cell and dendritic cell migration in perifollicular zone of lymph nodes. CCL21, the receptor is CCR7. The major function is T cells and dendritic cell migration into parafollicular zones of lymph nodes. CCL21, the receptor is CCR4. They are found mostly in natural killer cells and T cell recruitment. CCL25, the receptor's chemoreceptor is CCR9. They are found, or the main function is lymphocyte recruitment into intestine. CCL27, the chemokine receptor is CCR10. The main function is T cell recruitment in the skin. Then we have CXC chemokines. CXCL1, the chemokine receptor is CXR2, mostly found in neutrophil, and function is neutrophil recruitment. CXCL8 is mostly the chemokine receptor is CXR1, CXR2, and the main function is neutrophil recruitment. CXCL9, the chemokine receptor is CXR3. Their main function is effector T cell recruitment. CXCL10, their chemokine receptor is CXCR3. They are mainly found and function in effector cell recruitment. CXCL1, 2, CXCL12, the chemokine receptor is CXCR4. Their major function is B and T cell migration into lymph node, plasma cell migration into bone marrow. CXCL13, their chemokine receptor are CXR5. The major function is B cell migration to lymph nodes and into follicular T, follicular migration to follicles. We have C chemokines. These are XCL1. The receptor chemokines is CXR1. The main function T cell and the natural killer cell recruitment. We have CX3C chemokines. For example, CX3CL1, the chemokines is CX3CR1. The main function is T cell, natural killer cell, and monocyte recruitment. So these were just the example which you can easily follow. The receptor of chemokines belong to 7 transmembrane guanosine triphosphate binding G protein coupled receptor, the GPCR superfamily. These receptors initiate intracellular response through associated trimeric G protein. All chemokine receptor mediating immune cell migration share amino acid sequence and motif dry live at the end of 3rd transmembrane domain that is required for interaction with G protein. The G protein stimulates signaling event that result in cytoskeletal changes and polymerization of actin and myosin filament, resulting in increased cell motility. As previously discussed, this signal also increases the affinity of integrin for the ligand. Different combinations of chemokine receptor are expressed on different types of leukocyte, resulting in distinct pattern of migration of these cells. There are about 10 different receptors for CC chemokines, called CCR1 through CCR10. 7 for XCC chemokines, called CXCR1 through CXCR6 and CXCR8. One of the C chemokines, called XCR1, and one of the CX3CL1, called CX3CR1. Chemokine receptors are expressed on all leukocytes, with the greatest number on diverse seen on T cell. The receptor exhibits overlapping specificity for chemokine within each family, and pattern of cellular expression of this receptor determine which cell types respond to which chemokines. Certain chemokines receptor, notably CCR5 and CXR4, act as co-receptor for entry of human immunodeficiency virus HIV in cells. A distinct set of chemokine receptor called atypical chemokines receptor, ACR LACKRS does not engage in heterodynamic G protein signaling pathway that activate leukocyte, but rather is involved in inhibiting or terminating chemical response in cell. Therefore, there are 4 human ACKRs expressed on many different cell types that together bind most of the inflammatory chemokines within high affinity. One particular ACKR1 or DACR is found on erythrocyte and on the specialized endothelium of postcapillary venule, the site of leukocyte migration from the blood into tissue. ACKRS signal through a pathway dependent on the G protein regulatory protein called B-arrestin, which binds the chemokines receptor with attached chemokines and stimulate the internalization and degradation of these receptors. Function of chemokines. Some chemokines are produced by cell in response to external stimulus and are involved in inflammatory reactions. Other chemokines are produced constitutively in tissue and maintain distribution of cells in these tissues, such as localization of T and B cell in lymphoid organs. In inflammatory reaction, chemokines serve to recruit circulating leukocyte from blood vessel into extracellular site. Different groups of chemokines bind to chemokine receptor expressed on different cells and in coordination with these types of adhesion molecule expressed control the nature of inflammatory infiltrate. Chemokines play 2 roles in inflammation. 1. Increase the adhesion of leukocyte to the endothelium. Chemokines produced in the tissue bind to the heparan sulfate proteoglycan on endothelial cells that line postcapillary venules. The bound chemokines are displayed in this way to the circulating leukocytes that are attached to the endothelial surface through adhesion molecule interaction. Endothelial display provide a high local concentration of chemokines, enabling them to bind to chemokine receptor on leukocytes. Signal from chemokine receptor lead to the enhanced integrin affinity, which result in firm adhesion of leukocyte, a critical step for migration of leukocyte or out of blood vessel into extravascular tissue. Migration of leukocyte through blood vessel and toward the site of infection and damage is the second function of chemokine. Chemokine produced in extravascular tissue act on leukocytes that have migrated through endothelial and exited the circulation. Leukocyte migration toward the infected and damaged cell in the tissue where chemokines are produced at highest concentration. Chemokines are involved in development of lymphoid organs and they regulate trafficking of lymphocyte and other leukocyte through different regions of secondary lymphoid organs. Because these chemokines are expressed constitutively and maintain normal tissue architecture, they are referred to as homeostatic. We discussed the function of chemokines in anatomic organization of lymphoid organ in chapter 2. Some of homeostatic chemokines are also induced under inflammatory condition and contribute to leukocyte migration out of blood vessel in tissue. Chemokines are required for migration of dendritic cell from site of infection into draining lymph node. The dendritic cell are activated by microbes in the peripheral tissue and they then migrate to the lymph node to inform the T lymphocyte process of infection discussed in chapter 6. This migration depends on expression of chemokine receptor CCR7, which is induced when the dendritic cell encounter microbes and chemokine produced in lymphatic and lymphoid tissue that bind to CCR7. Naive T cell also expresses CCR7, and this explains how dendritic cell and naive T cell localize to the same place in lymph node, enabling the dendritic cell to present antigen to T cell. Other chemoattractant and receptor. Several other types of molecules serve similar function as chemokines in immune system, promoting leukocyte migration in inflammatory sites and their directed movement in tissue or maintaining spatial separation of lymphocytes in secondary lymphoid organs. The C3A and C5A complement fragment generated by activation of complement and the arachidonic acid metabolite leukotriene B4 TLTB4 are important chemoattractants that promote neutrophil and monocyte migration during active inflammatory response. Other lipid molecules play critical roles in the movement of leukocyte and dendritic cell in lymph nodes, spleen, and mucosal lymphoid tissue. These include sphingosine 1 phosphate, a lipid required for ingress of lymphocyte out of lymphoid organs, which is discussed later in this chapter, and oesterol involved in organizing lymphoid follicle and directing movement of B cell during gemino-center reaction discussed in chapter 12. All these chemoattractant molecules, like chemokines, bind to specificity GPCRs expressed on the leukocyte. Leukocyte endothelial interaction and leukocyte recruitment in tissue. Leukocyte recruitment from the blood tissue requires adhesion of leukocyte to the endothelial lining of post-capillary venules and then movement through endothelial and vessel wall into extravascular tissue. Leukocyte recruitment into the tissue is the key event in inflammatory reactions. It is a multi-step process in which each step is orchestrated by different types of adhesion molecules and chemokines. Study of the interaction of leukocyte with the endothelial in vitro under conditions that mimic flowing blood and in vivo using intravital microscopic technique have established a sequence of events common to migration of the host leukocyte into most tissue. The steps in this process are as follows. First, selecting mediating rolling of leukocyte on endothelium. Macrophage, dendritic cell, and other cells that encounter microbes in extravascular tissue are activated to secrete cytokine, including the tumor necrosis factor and interleukin-1. These cytokines stimulate endothelial cell lining post-capillary venules at site of an infection to express E-selectin endothelial cell also express P-selectin in response to histamine released from microbes activated mast cell, and thrombin produced during blood coagulation, which occur commonly in inflammatory reaction. At these sites, blood vessels dilate and blood flow slow. As a result, leukocytes, being larger than red cells, tend to move away from the central axle flow and closer to the vessel lining, a process known as margination. This allows the ligand for E and P selectin expressed on the microville of the leukocyte to bind to the selectin that have been induced on the endothelial cells. Because interaction of selectin with their ligands are of low affinity, kilodalton 100 micrometer, with a fast off-rate, they are easily disrupted by shear force of the flowing blood. As a result, the selectin ligand bonds repeatedly form and break, and the leukocytes are pushed along the endothelial surface in a rolling motion. This slowing of leukocyte on the endothelium allow the next set of stimuli in the multi-step process to act on leukocyte. Chemokines mediated increase in affinity of integrins. Chemokine displayed on endothelial cells of postcapillary venules at the infection site bind to their receptor on the rolling leukocyte. As discussed before, this results in stronger binding of leukocyte integrin to their ligand on the endothelial surface. Stable integrin mediated arrest of leukocyte on the endothelium. In parallel with the activation of integrin, the expression of the ligands on the endothelial cells is regulated by inflammatory cytokines and a microbial product. These ligands include VCAM1, which bind to the integrin VLA4, and ICAM1, which bind to LFA1 and MAC1 integrin. Thus, leukocytes attach firmly on endotheliums, their cytoskeleton is recognized and they spread out on the endothelial surface. Transmigration of leukocyte through the endothelium. Leukocytes usually migrate out of blood vessel between endothelial cells, a process called a paracellular transmigration or diapedesis, to reach the extravascular tissue. Paracellular transmigration depend on interaction of integrin on the leukocyte and their ligands on the endothelial cell, as well as the contribution of other protein, notably CD31, which is expressed on leukocyte and endothelial cells. This process requires a transient and reversible disruption of adhesion function proteins, primarily the VE-cadherin complex that hold the endothelial cell together. The mechanism responsible for the disruption of VE-cadherin complex involve activation of kinase when a leukocyte integrin bind ICAM1 and VCAM1. The kinase phosphorylate the cytoplasmic tail of VE-cadherin, which lead to the irreversible disruption of adhesion complex. Less often, leukocytes have been observed to move through endothelial cells rather than between them by poorly understood process called transcellular regression. These basic steps are seen in immigration of all leukocytes through endothelium. However, neutrophil, monocyte, and different subsets of lymphocytes differ in which tissue they migrate into and when they do so, an inflammatory reaction and the steady state. These patterns of leukocyte migration are dependent on the expression of distinct combination of adhesion molecules and chemokine receptor, as well as we'll discuss more in detail later. Evidence of the essential role of selecting integrin chemokines in leukocyte migration come first from antibody blockade studies and gene knockout mice, and then from the discovery of rare inherited human disease called leukocyte adhesion deficiency. An autosomal recessive inherited deficiency in CD18 gene, which encodes the beta subunit of LFA1, MAC1, and CD11C CD18 in the same cause of immunodeficient disease called type 1 leukocyte adhesion deficiency, in which there were marked defect in leukocyte migration and immune response. Patients who lack the Golgi GDP Fucose transporter need to express the carbohydrate ligand for E-selectin and P-selectin on the neutrophil have similar problem, resulting in a syndrome called the type 2 leukocyte adhesion deficiency. These disorders are characterized by recurrent bacteria and fungal infection, lack of neutrophil accumulation at site of infection, and defensive defense-dependent lymphocyte function. Rare human mutation in signaling pathway linking chemokines receptor to integrin activation also result in impaired leukocyte adhesions and recruitment into tissue and therefore ineffective leukocyte defense against infections, a syndrome called type 3 leukocyte adhesion deficiency LAD3. Migration of neutrophil and monocyte to site of infection or tissue injury. After maturing the bone marrow, neutrophil and monocyte enter the blood and circulate throughout the body. Although these cells can perform some phagocytic function within the blood, their main function, including phagocytosis and destruction of microbes and dead tissue cells, take place in the extravascular site of infection virtually anywhere in the body. Blood neutrophil and monocyte are recruited to tissue sites of infection and injury by selecting integrin and the chemokine dependent multi-step process, which follow the basic sequence common to migration of all leukocytes in tissue discussed earlier. As we will consider in detail in chapter 4, neutrophil are usually the most numerous types of leukocyte to accumulate in tissue within 24 to 48 hours after the onset of infection or injury, and are then replaced by monocyte. This probably reflects the fact that there are about many more neutrophil in the blood and they respond more rapidly to chemokines compared to the monocyte and other leukocytes. After entering tissue, neutrophils have short life span before dying by apoptosis, where the monocytes survive longer and may also proliferate into the tissue. However, in some inflammatory sites, neutrophils are not recruited at all, but monocytes are. This different migratory behavior likely reflect variation in relative expression of adhesion molecules and chemical receptors on neutrophil versus monocyte. Neutrophil expresses CXCR1 and CXCR2, which bind CXL1 and CXCL8, interleukin 8. The major chemokines with ELR motif that support the neutrophil migration to the tissue. L neutrophil recruitment is a consequence of an L and an abundant CXCL8 production by tissue resident macrophage and other cells in response to the infection. In contrast to the neutrophil, classical monocyte, which are the main types of monocyte recruited in inflammatory sites, expresses CCR2. This receptor binds several chemokines, most important for monocyte recruitment being CCL2. Thus, the monocyte recruitment occurs when resident tissue cells produce CCL2 in response to infection. Migration and recirculation of T cell. Lymphocytes are continuously moving through the blood, lymphatic vessels, secondary lymphoid organs, and non-lymphoid tissue, and distinct population of lymphocytes show different trafficking pattern through this site. When a mature naive T cell emerges from thymus and enter the blood, it home to lymph nodes, spleen, mucosal lymphoid tissue, and migrate to the T cells zones of this secondary lymphoid tissue. If the T cell does not recognize antigen in these sites, it remains naive and leave the nodes or mucosal tissue through lymphatic and eventually drain back to the bloodstream. Once back to the blood, a naive cell repeats its cycle of homing to secondary lymphoid organs. This trafficking pattern of naive lymphocyte, called lymphocyte recirculation, maximizes the chances that the small number of naive lymphocytes that are specific for particular foreign antigen will encounter that antigen if it shows up anywhere in the body. Lymphocytes that are recognized and become activated by antigen within secondary lymphoid organ proliferate and differentiate to produce thousands of effector and memory cells. These effectors and memory lymphocytes may move back into the bloodstream and then migrate into site of infection and inflammation in non-lymphoid tissue. Some effector and memory lymphocyte subsets are preferentially home to the particular tissue, such as skin or gut. The existence of different homing patterns ensure that different subsets of lymphocytes that are specialized to defend against different types of microbes, such as parasite, extracellular fungi, and bacteria, or intracellular bacteria, are delivered um, to the tissue where those microbes are, and not wasteful to place where they would serve no purpose. In the following section, we will describe the mechanism of pathway of lymphocyte recirculation and homing. Our discussion emphasized T cell because more is known about their movement through tissue than it is known about B cell recirculation, but many of same mechanisms appear to mediate the recirculation of both cell types. Recirculation of naive T lymphocyte between blood and secondary lymphoid organ. T lymphocyte recirculation depends on mechanisms that control entry of naive T cell from the blood into lymph nodes, as well as molecular signals that control the exit of naive T cell from nodes. We will discuss these two mechanisms separately. Migration of naive T cell into lymph nodes. The homing mechanism that bring naive T cell into lymph nodes are very efficient, resulting in a net flux of lymphocytes through lymph node that is estimated to be up to 25 times 10 power 9 cell each day. On average, every naive T cell in the body goes through at least one node once a day. Peripherally tissue inflammation, which usually accompanies infections, causes a significant increase in blood flow into the lymph node, draining the affected tissue and consequently an increase in T cell influx into those lymph nodes. At the same time, egress of the T cell into efferent lymphatic is transiently reduced by a mechanism we will discuss later, so that T cell stays in lymph node that drain site of inflammation longer than in the other lymph nodes. Proteins, antigen are concentrated in lymph nodes and other secondary lymphoid organ where they are presented to T cell by dendritic cell and types of antigen cell best able to initiate response of naive T cell. Thus, movement and transient retention of naive T cell in a secondary lymphoid organ, together with capture and concentration of antigen, maximize the chance of T cell activation and initiation of an adaptive immune response. Homing of naive T cell into lymph nodes and mucosa-associated lymphoid tissue might occur through specialized postcapillary high endothelial veins located in T cell zones. Naive T cell lymphocytes are delivered to secondary lymphoid tissue through arterial blood flow, and they leave the circulation and migrate into the stroma of lymph node through the high endothelial venules. These vessels are lined with the plump endothelial cells and not the flat endothelial cells that are typical of the venules. High endothelial venules are also present in mucosal lymphoid tissue, such as pear patches in the gut, but not in plasma and spleen. The endothelial cells of high endothelial venules are specialized to display certain adhesion molecules and chemokines on their surface, discussed later with the support of selective homing of only certain population of lymphocyte. Certain cytokines such as lymphotoxin are required for high endothelial venules development. In fact, high endothelial venules may develop in extra lymphoid sites of chronic inflammation, where such cytokines are produced um, prolonged period. Naive T cell migration out of the blood through high endothelial venules into the lymph nodes parenchyma involve the adhesion molecule L-selectin and LFA1 and the other chemokine receptor CCR7, which this process includes the sequential event described earlier for migration of leukocyte, but migration across high endothelial venules into lymphoid tissue involves particular adhesion molecule and the chemokines. Rolling of naive cell to a high endothelial venule in secondary lymphoid organ is mediated by L-selectin on lymphocyte binding into P nodes, and the high endothelial venules P node is sulfated 6-sil Lewis X carbohydrate attached to the glycoprotein backbone. The P node carbohydrate group that bind to L-selectin may be attached to different sialomucin on high endothelial venules in different tissues. For example, lymph node high endothelial venules, the L-selectin ligand is displayed by two sialomucin called glycan-bearing cell adhesion molecules, GlycCAM1 and CD34 in pair patches in intestinal wall. The L-selectin ligand is exhibited on glycoprotein called MADCAM1, which also is the ligand of alpha-4 beta-7 integrin. As with leukocyte migration in other sites, the subsequent film adhesion of naive T cell to high endothelial venule is mediated by integrin and mainly LFA1. The principal chemokine that activates naive T cell integrin to the high affinity state are CCL19, CCL21, which are uniquely involved in leukocyte homing to T cell zones, lymphoid tissue. The main sources of CCL19 and CCL21 is fibroblast reticular cell within the T cell zone, and CCL21 is also constitutively produced by high endothelial venules. These chemokines are displaced on the surface of high endothelial venules and recognized by rolling lymphocyte. Both these chemokines bind to the chemokine receptor CCR7, which is expressed at high level on naive T cell. This interaction of the chemokine with CCR7 ensure that naive T cell increase integrin avidity and are able to adhere firmly to high endothelial venules. Recall that CCR also govern dendritic cell migration with lymphatic into lymph node. The important role of L-selectin and chemokine in naive T cell homing secondary lymphoid organ is supported by many different experimental observations. Lymphocytes from L-selectin knockout mice do not bind to peripheral lymph node high endothelial venules, and the mice has marked reduction in number of lymphocytes in the lymph node. There are very few naive cells in lymph node of mice with the genetic deficiency in CCL9, CC21, and CCR7. Movement of T cell within secondary lymphoid organs. After entering lymph node, a mucosal lymphoid tissue, T lymphocyte and dendritic cell actively move in a way that maximize the chance for two cell types interacting with one another. Initiation of T cell mediated immune response requires that rare antigen-specific naive T cell recognize specific antigen displays on the surface of dendritic cell, which are also numerically rare. Although they their shared expression of CCR promote the localization of both these cells types to the same area of the lymph node or mucosal lymphoid tissue. There are though thousands of dendritic cell and naive T cell in this location. However, naive T cells are remarkably motile within the lymph node, moving like amoeba up to 12 micrometer per minute. T cell movement is stimulated by CCL21 binding to CCR7 on the T cell, FRC that secretes CCL19 and CCL21 form three-dimensional network that traverse the T cell zones of lymph node and T cell zones along these FRC track. Dendritic cells are also distributed along the FRC network, covering most of the surface, and although they are not highly motile, they constantly extend their dendrites in different directions. As a result, each dendritic cell can contact many T cells over time, as many as 80 per minute. A single naive T cell may move along the FRC network in one lymph node for up to 24 hours, and therefore, if there are some dendritic cell displaying a particular antigen in lymph node, a naive T cell specific for that antigen that enter the nodes will likely find one of those dendritic cells. Immediately after recognizing antigen on dendritic cell, the T cell stop moving and the interaction with dendritic cell is stabilized, which allow full activation of T cell. Exit of naive and effector T cell from lymph node. Naive T cell that have homed into lymph node but fail to recognize antigen and are not activated will eventually return to the bloodstream. This return to the blood completes one recirculation loop and provides the naive cell another chance to enter the secondary lymph node organs and search for antigen they recognize. And the major route of re-entry into the blood is through the efferent lymphatic, perhaps via the lymph node in the same chain. Then through the lymphatic vesicle to the thoracic duct, which drain into the superior vena cava, and the right lymphatic duct, which drain into the right subclavian veins. The egress of naive T cell from lymph node is dependent on a lipid chemoattractant called sphingosine 1-phosphate, which bind to GP Pizarra family receptor T cell called sphingosine 1-phosphate receptor. SIP is present at higher concentration in the blood and lymph than in tissue. This concentration gradient is maintained because SIP degrading enzyme SIP-lyase is present in most tissue. So lipid is catabolized in tissue more than in lymph node and blood. SIP stimulating migration of cell toward the gradients to SIP circulating naive T cells have very little surface SIP because their higher blood concentration causes interaction of the receptor. After naive T cell enter the lymph nodes, where the SIP concentration are low, SIP appear on the cell surface over a period of several hours. This time lag allows the naive cell to interact with antigen-presenting cell. If naive T cells does not recognize any antigen before the SIP receptor is re-expressed, the T cell leaves the lymph node and is directed down to SIP concentrating gradient into the efferent lymphatic and eventually return to the blood. The egress from the lymph node of effector T cell generated from antigen-activated naive T cell is also dependent on SIP, where the naive T cell is activated by antigen in lymph node. The surface expression of SIP is suppressed for several days, and thereafter the ability of the cell to leave the lymphoid organ in resource to a SIP gradient is delayed. This suppression of SIP is controlled in part by cytokine called type 1 interferon that are produced during innate immune response to infection, as well as we'll discuss in chapter 4. Antigenic stimulation and interferon together increases the expression of protein called CD69, which bind to intracellular SIRP and reduce its cell surface expression. Thus, the activated T cell become transiently insensitive to SIP gradient, which shows the antigen activated T cell to remain in the lymphoid organ and undergo the clonal expansion and differentiation into effector T cell, a process that may take several days. When differentiation into effector cell is complete, the cells stop expressing CD69 and therefore SIRP again expressed on the cell surface. At the same time, the new regenerated effector cell reduces expression of L-selectin and CCRS5, 7, which previously attracted the naive cell to lymph node. Therefore, the effector T cell become responsive to the constraining gradient of SIP and exit lymph node via the medullary sinus, draining into the efferent lymphatic and eventually re-enter blood. SIP and SIP1 are required for mature T cell ingrained from cymath and migration of B cell derived plasmablast from secondary lymphoid organ. Our understanding on the role of SIRP and SIRP-R1 in T cell tracking is based on part of studies of drug called fingolimod, which bind to SIRP-R1 and causes its downregulation modulation from cell surface. Fingolimod block T cell egress from lymphoid organ and thereby act as immunosuppressive drug. It is approved for the treatment of multiple sclerosis, autoimmune disease of the central nervous system, and use of fingolimod and other drug with similar mechanism of action to treat other autoimmune disease, and graft rejection is under investigation. Additional experimental evidence for the central role of SIP in naive cell trafficking comes through studies of the mice with genetic aberration of SIRP-R1. In these mice, there is a failure of T cell to leave the thymus and populate secondary lymphoid organs. If naive T cell from SIRP knockout mice are injected into the circulation of other sites, the cell enter the lymph node but are unable to exit. Today I'll end up here and we'll continue where I ended up.